Well, welcome to Wave Club, everybody. Happy Monday. I am Josh Jacobs here in the bottom left with the outdated photograph of myself. And Wave Club is organized by Bard Ehrmantraut, the second person there, Uma Mohan, and Irfan Zabeh, who are all here to helping me organize this wonderful event. We realized we've been doing this for almost 2 years now, and we have a bunch of older Wave Club recordings on YouTube if you're interested to check them out. The purpose of Wave Club is to be an interdisciplinary event that brings together neuroscientists from all across neuroscience, humans, animals, computational, empirical researchers, to study the phenomenon of traveling waves, because this is an emerging field that's been really growing a lot over the years. Our presenter today is Sage Chen from NYU. My next slide.
Yeah.
Awesome. Great. Here is Sage in a very nice photograph here, who's going to be giving a talk today on traveling waves as a communication scaffold in structured brain networks. Um, Next slide, please.
Yeah.
Sage is a professor in the departments of psychiatry and neuroscience at NYU. He has a PhD from McMaster University and did a postdoctoral fellowship at Harvard and MIT. And he at NYU, he leads the CN3 lab, studying computational neuroscience. And I'm familiar with, so here's a more formal listing of uh, Sage's work. He does a lot of work on both computational modeling with as well as empirical studies of different kinds of network structure and neural networks, ranging from animal studies and including new work in humans that I believe is going to show us some more today. Um, and he also published a new book that is coming out very soon, uh, from, I believe, MIT Press. So that's very exciting as well. Um, Next slide, please. Um, so before we get started with Sage's talk, here's a link to our YouTube channel, which is youtube.com/@waveclubseminar. And you can also follow us on Blue Sky and also Twitter, @waveclubseminar is our username, um, to to get more of a sense of to learn more about our work and stay in touch with our meetings, which happen about every month. Sage is probably going to present for 40 minutes today, something like that, leaving time for questions. The best way for us to have conversations is for you to come on stage at the end of the, virtual stage at the end of the meeting. We can have a conversation about your questions with Sage. Um, and if you want, you can tap your question. But if you prefer, you can tap your questions in during the talk, and we can review them while things are going on. And I believe Sage mentioned to us that he's going to have two different talks, two different parts to his talk, with a split halfway through. So we might have a question and answer session halfway through today, which is just great. And before we get started, talking about Sage's work, I wanted to remind you that every month we have a different speaker for Wave Club, and we are always looking for new speakers going forward. So if you are interested in presenting at Wave Club, feel free to drop me or one of the other organizers an email or a DM on Twitter or Blue Sky, so we can try and organize a schedule and possibly have you talk, because we're really interested in having this be a diverse, welcoming group of people giving presentations at different career levels and things like that. So feel free to be in touch if you're interested in speaking or you have a different suggestion for us for somebody else to be a speaker. I'm going to be quiet now and turn the stage over to Sage. Irfan and Uma and Bard, thanks for helping me organize this today. Appreciate it. And Sage, thank you for joining us, of course.
Thank you. Let me just share the screen.
Sage, that's a wonderful background with
Oh,
your whiteboard's gotten a lot of use there. It's an excellent Zoom background. I'm impressed.
yeah. Well, thank you. Thank you everyone for joining up this wake up presentation. And I'd like to formally thank Uma, Efran, and George and Bar for inviting me, giving me the opportunity to talk about this actually unpublished work in front of this audience. Let's see. So the outline of the talk are following. So, for this audience, I would, briefly give a background on traveling wave. I don't think I need to spend too much time talking about it, given, the audience who already many of them are expert in this domain. So my talk primarily consists of two parts. Part one is basically, so actually these two stories, actually these two parts actually are done by former undergrad student on my lab. This is all non-published. It's really driven by their, summer intern project in a sense, and look into those particular problem. So part one is focused on data. So this is a focus on a data set that collected from Michael Kahana's group at UPenn. We're looking at hippocampal and parahippocampal tramway based on human intracranial EEG recording during memory processing and memory recall. Second part of the talk, I switched the gear a little bit, focus on a little bit modeling. And we try to do modeling for there's a multi-region hippocampal and also hippocampal cortical tramway. I also touched the base a little bit of the whole brain tramway project at the very end. So obviously these two parts are relatively independent, but I try to sort of like, get to the, in the discussion, and I try to, reach the point that basically to modeling, to understand this tramway phenomenon is important to looking at multiple brain area, and the modeling, and given the technology, so the limitation this day is not that common to simultaneously detect and record those, a mesoscopic tramway at, multiple brain region, right? So modeling will provide sort of mean, a complementary mean to looking at tramway. So the goal of the modeling, obviously, threefold. For example, you try to make sense of the data. If you have the data collected already, try to make sense of the experimental data. Second is try to test the model assumption. So you made some assumption in the model. So you really want to see whether those assumptions are correct or robust. The third is actually is try to make experimental predictions as well, because many data has not been collected yet. So you can presumably perturb the circuit or perturb the model to make prediction. So I will conclude the talk at the end, so with some discussion and outstanding question. So today, you probably, for this audience, I don't have to convince you. There are many paper and many review article has been written, published on the topic of the tramway. And tramway, the notion of the tramway is not really new. It started from physics, and then, decades ago, it started to become more popular in neuroscience community, right? So people talk about, mostly focus on the cortical tramway, because the recording allows us to detect those macroscopic and mesoscopic tramway. So there are many sort of angle being written, so I won't go into those. So people with interest reader, you can sort of looking at those review. Meanwhile, you can also see the number of publication on tramway also growing. This is a slide taken from a recent review article from Eli. You see that, people talking about at the global brain level or macroscopic level, or in the circuit, the column or area level, which is a mesoscopic level, right? The number of publication has growing almost exponentially, every year. This is only up to 2020. I believe in the last 5 years, there are more publication has been appeared in the field. Meanwhile, people also interested in modeling. Pioneer Barr here, as one of the co-organizer, is the expert in this field, has been modeling tramway, decades ago, starting with decades ago, right? So the modeling, as I say in the beginning, that would provide some sort of a complementary tool to looking at tramway phenomenon. So this is just give you sort of a very, very sort of high level background that, tramway, especially spontaneous tramway, they call tramway, are really ubiquitous feature in a different brain state, a different circuit, different brain region, at different spatial and temporal level, right? So in the left, you can see that basically this is, I think, a slide recorded from a weight marmoset, I believe, in MT region. You can also see that using the 2D array, Utah array, or using other basically array, 9x9 array, to detect this LP, sort of a tramway phenomenon. I believe people also reported in a motor cortex based on mesoscopic LP recording as well. Panel B showing that, in contrast, showing this macroscopic spontaneous tramway in the whole brain, right? In the human, people typically relies on resting state fMRI. There's no task, just really the spontaneous activity. Looking at both bottom-up propagation and top-down propagation, you can see that there's a sort of a pattern, this wave pattern propagated through this cortical, sensory motor, or transmodal hierarchy in both bottom-up and top-down direction. Not only in the human fMRI, but also this phenomenon also being observed in the monkey ECOG signal as well. In panel C, what we see here is actually an interesting figure that from this neuron review. It was showing that basically there's a global coordination way follow this causal periodic pattern that kind of closely approximate this cortical hierarchy. So if you look at the figures, this is you can see that based on the fMRI data, so they see this resulting pattern activate with a global cycle in coordination with the subcortical structure, which is showing this bar charts. Each beam of the it shows the number of the voxel activated at each time point for each brain structure, and the color match the activation order of the voxel show on the cortical surface, showing above this figure. So it's very interesting. The subcortical structure also showing there's some sort of temporal order in this activated pattern. So this is a slide taken from Yuri Buzakis' lab. So in the Buzakis' lab, they actually recorded simultaneously using wide field calcium imaging and electrophysiology. In this case, they implanted electro recorded the rodent brain. This is a half-fixed mice in electrophysiology recording implant on the retrospinal cortex as well as the hippocampus. And they also using wide field calcium imaging to record, half hemisphere about the whole brain, right? So there's a mixed recording, and the concurrent recording give them this sort of opportunity to look into this spatial temporal, you can call it wave pattern, how this pattern change conditional on the hippocampal shower ripple, for example, right? So, for example, looking at once you detect the hippocampal shower ripple, how this tramway occur, what's the onset, which region started early on, and then when did they, how did they propagate in terms of direction, what's the relationship relative to hippocampal ripple. So, and now we'll talk about a little bit about a communication scaffold. What do we mean by communication scaffold? And we know that biological networks are structured, especially modular, right? If you talk about a different level, and then and the brain is not really random in terms of connectivity. When you especially talk about a neighboring structure, upstream and downstream structure, right? So if you believe this principle structure primary determine the function, right? If you have this structure and there's a reason based on the evolution or for other reason, computational reason, the function is really, determined by this structure. At the macroscopic level, we talk about the scaffolding is really determined by the structure backbone, right? So you cannot really, you actually found that the functional connectivity is really resembles some sort of a structure information, right? At the microscopic level, on the other hand, the scaffolding is really determined by the synaptic connectivity between neighboring neuron and how does synaptic connectivity would determine the function as well as the dynamic and pattern and so on. When we look at the traveling wave, so the traveling wave, so essentially it's really nothing but a smooth change in the phase of the neural oscillation or event-related response propagated in time and space. Right, using the example here, Josh's work published in 2018. Right, this is talk about hippocampal traveling wave. So you can see that hippocampal theta, after you filter the the intracranial EEG signal, you can see the filter data. There's a different electrode, you see a consistent phase shift, right? This is only possible when you have like a larger scale the electrode implant in one brain or multiple brain region. So I think in the past people haven't really paid much attention, and since there's a recent sort of theory modeling on this traveling wave, many people started to pay attention on their recording, all right, and they reported many sort of phenomenon as an emergent property of the brain. When we talk about the traveling wave, sort of interesting to think about the different category, right? So in this review, the Eli paper reviewed, they introduced this first order traveling wave notion versus the second order traveling wave notion. So the first order traveling wave is referred to this propagation neural activity, if you like, between interconnecting neurons. Essentially talk about kind of a microscopic level. There's a synaptic connectivity. And the second order traveling wave phenomenon is really referred to a little bit more large sort of a spatial scale. So this is looking at a propagating pattern that result from this interplay between different multiple individual module that can include feed-forward module and feedback module within a very large network, right? So this is talk about different sort of spatial scale. So in terms of function, traveling wave has been suggested like play important role in information transfer, and memory maintenance, and neuroplasticity, right? So there's a different sort of depends on the task, depends on the brain state, they can presumably play different roles, at least for facilitating of those functions. So, and we talk about this communication, neural communication between two brain area. And we talk about two brain areas, the upstream structure and the downstream structure. There are different theories has been developed in the field, arguing how the neuron, how the brain area communicate information or have this communication between each other. One of the theory, which is called communication subspace, right? This is really focused on a little bit on the cellular level, if you like. So basically, the theory is saying that, if you're looking at upstream, downstream structure, there's subset of the neuron, right? Dedicated their effort only talk to another subset of the neuron in the other brain area. So between the communication, they form communication subspace. This is using a probabilistic method called canonical correlation analysis to looking at the communication subspace. And the theory has been demonstrated in many systems, including the visual system between V1, V2, and also in the recent work in collaboration with Buzak's lab, we showed that it also occur in the hippocampal CA1 and CA3, as well as the retrosplenial cortex as well. So this is what happened at the cellular level. Spiking resolutions is very different, different spatial and temporal scale. The other theory is based on synchronization. This theory is also not new. Has been proposed, has been existing for many decades, right? So the idea is that between two brain areas, they can use in a rhythm or oscillation to communicate, whether it's using coherence or using a nasty oscillation. So there's a different theory. Try to focus on slightly different aspect of this communication. I would say third, the theory would be traveling wave, right? So I think Terry Sinowski and many others also proposed that traveling wave could be a very useful tool. If it's not the only one, it could be a useful tool to communicate and send information across the brain. So, and this is the for his unique sort of feature. The question is really like how, right? So asking if you believe this is a reasonable hypothesis, a reasonable theory, so the question is the how, and what property of the traveling wave matter, right? So if you think about the communication matters, you have to look at the property in the context of tasks, right? So many of the phenomena is really happen in spontaneous state. People looking at a tremor wave pattern, the spontaneous state or resting state. So those things without the context, the task is harder to interpret or really this ambiguity of the role of the tremor wave. So also, technically, in order to answer the question, so it will require us to, recall multiple brain area, so multiple sign brain recording, presumed and hopefully using electrophysiology, right? Because the better temporal resolution, and also detect simultaneously the tremor wave phenomenon at those two areas, downstream and upstream structure. They impose some challenging because, in the past, it's not really possible. And recently, because the larger scale neural recording become more popular, you see NeuroPixel and other high density array recording. So this become technically at least feasible. So today we're going to focus on areas so hippocampal-entorhinal cortex. This hippocampal-entorhinal system has been implicated in memory and navigation. This is for example, in this review has been discussed about the theta rhythm in the context of this what the role of the theta will play in memory and navigation in this system. If you look at this anatomy structure, looking at the comparison between rat and monkey and human, you can see there's a very similar structure in this hippocampal-entorhinal sort of system, right? So in the rodent and rat, you can see there's a bell shape, the hippocampal structure, like from dorsal to ventral. This is typically in rodent, the dorsal to ventral structure in the bell shape, hippocampal. And in monkey and human, so and primate, typically this is we don't use in the dorsal ventral terminology. We refer to the posterior-anterior structure. So when we talk about dorsal in rodent, that map to the human in or primate in the posterior to anterior, right? And the entorhinal cortex and hippocampus, they are neighboring structure. The entorhinal cortex presumably provided the input projected to the hippocampus. Is showing the figure in the top at the bottom left, right? So you can see that hippocampus have a different subfield, right? The MEC or medial-entorhinal cortex send the input to dentate gyre and the CA3 and the CA1. This like a trisynaptic loop. They also MEC also provides some direct input to the CA3 as well, right? So this is a little bit overview about this hippocampal-entorhinal cortex system. I'll give you a little bit context. So in the part one of my talks, I would basically dive into the this human ECOG, intracranial EEG recording. Data has been collected from my Kanhana School in UPenn. It's public available. People, are welcome to explore it. That's really motivated one of my former undergrad trainee to look into the data during his summer internship. And this data set has been very rich. Has been, many high-profile papers have been published, right? So you see, I just highlight a few of them here. Many of them co-authored by Josh. So you can see that in the top right. So this is the basically showing the hippocampal theta. So the tremor wave showing in the electro, you see a consistent phase shift. In the bottom, so this is recent publication by Josh's group showing that there's theta and alpha sort of a tremor wave pattern during the same task phenomena we described here, but this is focused on more cortex based on the array structure. Right. So we basically taking the same sort of we just sort of like looking for structure, right? So we say, okay, there's some publication out there, published literature there. So why don't we just looking at something maybe joy sort of concurrent pattern, right? We focus on not just on one region, we're presumably looking at both regions and looking at the coordination between these two regions. So one of the reasons we're looking at hippocampal and parahippocampal for obvious reason, because this two regions that belong to the human medial temporal lobe, So we sort of have a good sort of argument to say these two areas are important for information transfer, for memory processing. So the parahippocampal gyrus consists of entorhinal cortex, also perihedral cortex and parahippocampus. So because the electro implant in each epilepsy patient are very different, so we basically limit to subset of the subject that we can reliably detect have a sufficient sort of implant coverage for those two areas, because we really want to looking at the both area, right? So the question we really try to ask in this investigation is really following. So what's the role of the traveling wave in memory encoding, right? Especially when we talk about two regions, right? Hippocampal and parahippocampus. Do they play similar role? What was the sort of coupling between them and so on, right? So it's really comparing sort of a task performance in this free memory recall, successful trial and unsuccessful trial. So we basically took a similar approach, like any other traveling wave paper, and detect the traveling wave, with this pre-processing. The detection procedure is really standard, so I don't want to go into the detail. It's essentially after bandpass filter, and then we basically fit a line or a plane, if you're allowed to fit a 2D plane, fit a 1D line or 2D plane to detect the traveling wave propagation direction. We see that here, showing you some example, which is on the left is entorhinal cortex traveling wave. So this is, we can see example in both posterior to anterior direction as well as the anterior to posterior direction. On the right is actually a hippocampal structure, traveling wave. So you can see that this is also we see posterior to anterior as well as anterior to posterior direction. These two directions, we didn't see any sort of a dominant pattern, which direction is more dominant than the other. It depends on a different task phase. Essentially, we see that this kind of a omnipresence is always present across different task phase. So next, we're also looking at the traveling wave, so like what was the sort of relationship in terms of the functional connectivity. In this case, we have the possibility to examine the functional coupling connectivity between left hemisphere or right hemisphere if the implant allows. So in some cases, we can detect the intra-hippocampal, sort of a left versus right hippocampus. Some cases we can look at between hippocampus and parahippocampal functional connectivity. We look into a couple measure and two measure. I want to highlight here. One is the phase locking value. This is looking at more phase, so the coupling. The second is for coherence, and this will talk about mostly amplitude, amplitude coupling at both theta and low gamma bands. We're also looking at other bands, but I think only these two bands kind of stands out and also can relate to the literature in terms of interpretation. We didn't find any sort of significant pattern in the other frequency band. So first we see that, traveling wave, once we detect the traveling wave, we're looking at this, what happened in the traveling wave during this window compared to outside the traveling window. And we see that the traveling wave can elevate this phase locking value and coherence quite significantly. We also see that traveling wave also elevate this theta phase and gamma amplitude coupling. This phenomenon, I think, is not really new. It has been shown in previous publication in Kahana School and some of the publication by Georgia School. So I think it's just really confirmation. So this is not surprising per se. The surprising result we found in this investigation, we see that hippocampal theta is narrow band between 5 to 9 hertz, as well as the low gamma defined by 35 to 55 hertz. The power by looking at comparison between successful versus unsuccessful trial, we see that the successful trial are lower, right? This in the beginning, we think, oh, this is a little bit interesting, but a little bit surprising. We thought, like counterintuitively, right? So we thought, oh, maybe the successful trial require more power, but it's actually the opposite, right? So it's actually the successful trial require less power. So then we look into literature and then see whether that makes sense, any sort of evidence support that. So we actually found that in the early work by Kahana's group, showing in the bottom, the neuroimage paper, they actually report similar phenomenon. They actually looking at hippocampal and parahippocampal structure, and they reported this reduced raw theta power in the sort of a wide range of the area, but only in the frontal brain area they show the increased theta. So this is showing that you can see one of the figure on the right, in the right top right, right? They show this z-score power, right? In the theta band, especially in the hippocampal and parahippocampal, they show this gamma power decrease, right? We report a similar phenomenon because this is really based on a simultaneous pattern, that we're looking at subject have a bow, hippocampal and parahippocampal structure. And we see that theta power significantly reduced and gamma power, the amount of reduction is a little bit less because the gamma power itself is lower than the theta, but it's still significant. So the interpretation we have for this finding is that, also by looking at the literature, we speculate that the spectral power reduction may reflect this active desynchronization, right? So there's a debate what's the role of the synchronization and desynchronization and their role in episodic memory. So I think that kind of supporting the early sort of claim that spectral power reduction reflect active desynchronization and facilitate this more efficient sparse neural coding. This is obviously still a speculation hypothesis level, so they require further investigation. So then we also look into sort of like the information flow between the region, right? So, and one of the measure we use is using granular causality, right? This is a measure sort of looking at the predictability, like from, the history of the one information to predict the information of the other region. So, and we're looking at using basically looking at both trial average and moving window kind of a granular causality analysis. And we're looking at both direction between hippocampal to parahippocampus, right? Both direction. And cross frequency, you can see that there's a strong sort of information flow from parahippocampal to hippocampal. This is sort of supporting the input-output, right? Hippocampal, sort of a parahippocampal and entorhinal cortex, the services of the input, propagate information to hippocampus. Interestingly, this phenomenon is quite consistent across different task phase. Like you see the countdown, which is really has nothing, it's sort of treated as a baseline, but also in the encoding and the retrieval phase. We don't see a significant difference in terms of the profile. And then we're also looking at particularly looking at encoding stage, and then we're looking at between task performance. And this is really comparing the granular causality strength and between successful try and successful try during the encoding phase. And we see that there's some sort of significant difference in unsuccessful try compared to the successful try. Our interpretation, again, here is a little bit sort of surprising. So we see that the unsuccessful try seems to exhibit more sort of excessively actual information flow, and they're acting like a neural traffic jam. So this is one interpretation. So there's so the interim discussion here. So we believe that tramway can act as like a spatiotemporal highway that facilitate information transfer across medial temporal different structure, hippocampal and entorhinal cortex system. And successful memory recall is associated with the reduced gamma crosstalk. Presumably, they will help to improve the signal-noise ratio and improving the redundancy, right? Obviously, this hypothesis requires EFIS recording to confirm if this is correct, right? We also come up with the sort of based on our finding, we come up with the second hypothesis: the tram wave can act like a gate control, a gatekeeper, right? Try to minimize the interference to facilitate this high quality neural communication, right? One way to looking at to test this hypothesis is that you would detect or at least recall so the hippocampal, cortical, hippocampal, parahippocampal, cortical tram wave to see whether there's any coordination between this tram wave pattern. Interestingly, I just want to bring people's attention that Josh's group have one I think paper coming up that sort of looking into insular structure of the tram wave. Also, they see some sort of differences between tram wave pattern can allow them to detect successful trial and unsuccessful trial during the encoding, and the tram wave strength also can show some differences at different frequency band during the encoding phase. And this is interesting because we know that hippocampal and insular structure, there's some direct interaction highlighted by the paper recently. It is Nature Neuroscience paper. I strongly recommend people look into it and looking for the sort of interpretation how this hippocampal and insular structure, the tram wave pattern are related, right? So this is the sort of first part of talk. So I don't know if people have any question. If not, I will just move on to the second part of talk, which is focused on, I switched the gear a little bit, talk about a little bit rodent work and then modeling, right? So, and I don't have to repeat. So hippocampal CA1 theta tram wave has been, well known in the field that basically motivated a lot of study we talk about here. So this is work that is done by my former undergrad trainee. Now she's a grad student at Stanford. So
hey Sage.
Yes.
Yeah, there was one question in the chat.
Sure.
Did you want to take questions now, or do you want to wait till later?
Yeah, I could. Yeah.
There was a question from Dan Hess again. How does the oscillatory power relate to the how does the power relate to the amount of traveling waves? Do they closely relate? I think that was the question.
So, I mean, we look at the power, essentially we focus on one way is that we're not exclusively looking at the tram wave per se, we're just looking at the general power. So when you have a tram wave itself, tram wave obviously, detect the tram wave in theta, so the theta power obviously is more pronounced. But I think in students' work, I think he mostly focused on when we made the claim about the power reduction, it's not restricted to a tram wave pattern. So it's actually extended to all the region, all the area, including the one that without tram wave. Because I think the reason we took that approach is because the tram wave is a detection issue, right? So based on your implant and based on your array structure, how do they? So it's a detection issue. So we don't want to limit only tram wave detection and link it to the power story. I think the power phenomenon is not limited to the tram wave per se, because the tram wave detections only happen in a subset of the duration during the whole recording. Does that answer the question? Okay, so I will move on. So basically, this is the word modeling word just try to replicate the literature, essentially looking at a simulated behavior animal. A virtual animal running in the circular maze, and then with the default run speed, and then we can see this dorsal ventral sort of this axis, the tram wave propagated in this video. Without going too much detail, just give you sort of a highlight about this word. Essentially, we're looking at modeling to looking at this, taking a lot of information from anatomy, from in the literature in terms of the size of the hippocampus. We looking at sort of think about building different block of the in the hippocampus. And here, because the assumption is that hippocampus have a recurrent structure, so we're making the assumption this is happen the model really try to replicate CA3. And then because CA1, there's a less sort of a very sparse recurrent connection, especially in the E cell, EE connection. So we sort of we envision this model will first happen in the hippocampal CA3, and this information tramway can propagate to the CA1. And this we in each block, and this 12 by 5 block is really just motivated by the anatomy, so the size of the hippocampus. And then we also consider different kind of combination between exciting neuron using the leaky integrate firing neuron and inhibiting neuron. We vary the ratio as well. And in one version of the model, we have about 3,600 neurons. And very briefly, so this leaky integrate firing neuron. So we consist of exciting neuron and inhibiting neuron. So exciting neurons just assuming there's some place cell tuning, right? So with a particular center in the position, and they have the receptive field size. The receptive field size will carry some structure. I'm going to show you in the next slide. The inhibitory cell is a theta that's driven by some sort of receive input from septum, median septum, and then also there's additional run speed modulation that control the gain of the firing rate. Right. So as I say, there's an assumption we made in the model. So we're showing that based on the literature, we're showing the model, each place field around the axis of the longitudinal axis, there's an increasing receptive field size. Also, we also assume that the magnitude of the excitatory spatial input and the inhibitory theta oscillation drive, the amplitude or the magnitude would decrease as well around the dorsal ventral axis. Between the neighboring block, we made different assumptions. There's a long range reciprocal but asymmetric, and so the connectivity. The connectivity could be excitatory or inhibitory. But we found that by investigating two-part option, we found that inhibitory connectivity is more robust or stable, make the tramway more stable, right? So this is just the schematic. It just shows there's a spatial input, theta oscillation driving the exciting neuron and inhibiting neuron, right? And then we have the connectivity, synaptic connectivity between those block in both longitudinal direction and transversal direction. So after that, we basically play around those parameters. We are able to, producing this two-dimensional, so the tramway wave phenomenon shown in the video. So we can quantify this tramway speed. We can also, looking at the particular pattern related to firing, for example, and then we, some of the quantifications related to the delay in the phase shift, into the delay, whether it's absolute delay or relative delay. We also vary the ratio between exciting neuron and inhibiting neuron, see how the tramway phase shift is robust across different assumptions, vary the run speed as well to see that the run speed modulate the firing rate, but also is robust respect to the tramway sort of a phase shift. This is the purpose of the modeling is a little bit is hand tuning, the parameters hand tuning. We try to get the model to match the experimental data as close as possible, obviously, right? That's sort of the goal. We also look into not only run behavior, we also try to model the phenomenon during sort of REM sleep. We also see REM sleep have a theta oscillation. So then, we randomly select some of the exciting neuron, about 15% of the cell acted, and then, try to basically, in this case, there's no spatial input during sleep, only sort of internal structure. And we're able to replicate this tramway phenomenon as well, although the phase shift, so the mean is kind of shifted a little bit, and the variance is higher. This is understandable. So this is try to match some of the data, published data in the literature as well. And furthermore, since we have the neuron firing, so we can look at those neurons, how do they fire respect to the theta phase, right? So we see that, this is a cartoon, not cartoon, it's actually simulated data. This is the 3 example of the neuron. They showed that, overlapping receptive field, but it's a different sort of a center. And you can see that there's some theta sort of phase perception. You also see that the neuron shows some sort of prefer firing in the local theta in the trial, right? So even those neurons without they might not have a direct synaptic connection. So with this trialing way, because they fire locally, prefer locally at the theta trial, so you can imagine that those neurons can communicate or coordinate the information through this relative theta phase shift, right? This is we're talking about between 0 to 180 degree from dorsal to ventral axis. This is equivalent to about 60 millisecond, right? So if you try to linking this sort of a story to the STDP, so this presumably give you a way using this firing, co-firing activity, presynaptic firing, postsynaptic firing. If they have a long-range connectivity, you can promote or suppress this synapse, long-range synaptic plasticity. We also look into make some prediction, right? So for example, we see that since we have the LP, so we can looking at the coherence, the theta coherence at different regions of the hippocampus, right? And so we see that our model kind of match the experimental data that's showing that a different parameter layer coherence modulated by different run speed. So you can see that in the ventral part of the hippocampus, they're showing higher coherence between, so the more reddish compared to the dorsal part of the hippocampus between the channel. Right? This is to match the experimental data. So why do we care about this theta, right? So one argument is that whether this theta will provide some sort of behavior readout, right? In the literature, I mean, there's a couple paper has been showing that this relative hippocampal theta phase can provide a very reliable behavior readout. Essentially, the theta itself is actually provides rich information for reading out the animal's position, right? This has been shown early work in the Buzakis lab and also recent work by the same group, by Sommer's group at UC Berkeley. We also have so the follow-up work as well, published in sort of follow up the same sort of a line of thought, published in a few years ago. So after that, we sort of like take one step further to say, okay, now we really look into sort of like upstream, downstream structure. We know the entorhinal cortex, hippocampus, have a synaptic connectivity. So the difference between entorhinal cortex, hippocampus is that the place cell in the hippocampus have place cell that's modulated by the place, and entorhinal cortex, which just have the grid cell, kind of have this grid-like firing property. And we know that there's a strong anatomy connectivity between entorhinal cortex, MEC, to the hippocampus, right? So we will modify our model structure, try to really replicate some of the phenomena we see in the literature to the medial-entorhinal cortex. In this case, we can looking at both source and the target by change the connectivity pattern between the entorhinal cortex and the hippocampus. MEC serve as an input or source, and the hippocampus, in this case, will be CA3, will be served as a target. And so we can see that there's some coordinated sort of a pattern in the trial way. Then we, I think this is quite robust, obviously determined by the connectivity pattern, but it's relatively robust. Yeah. So then we can further look into between two regions. There's a theory about this entorhinal cortex, hippocampus. There's a global theta. Then suppose the global theta is coherent. We want to test whether in our model that would support that claim. We do see that at least qualitatively, we see some strong sort of theta coherence. Looking at the different dorsal to dorsal, ventral to ventral, different sort of anatomy structure. Relationship to the coherence. This is definitely stronger in theta, definitely stronger than the gamma. So do we see qualitatively similar phenomenon compared to the experimental finding, right? So for this part, so I would, the interim discussion is that trauma way facilitate the coherence within the hippocampus, also between hippocampal and entorhinal cortex, this between region communication. In our model, we see that there's an interconnectivity in the model really determine the tramway pattern. But obviously, it's sort of we try to the assumption we made in the model, try to match the literature, but also we found that with the right range of the parameter set up is quite robust, right? But it still depends on the parameter you made in the model. And then we found there's a long range. Remember, between block by block, we need to show this long range sort of connectivity. We found that the inhibitory long range inhibitory connectivity is more robust for tramway generation and tramway pattern. So obviously, our model made simple assumption, right? First, we assume that the cell property drive the tramway, right? It will have exciting neuron, sort of like spatial driven. And then we have inhibiting neurons, sort of theta driven, right? We're showing that those property, determine the tramway generation and propagation. But meanwhile, the tramway can, in our model, we didn't really assume the tramway reshape the cell property or synaptic strength, right? Essentially, our system is an open loop system, and we didn't really think about the other direction, right? Also, when we model the MEC, this CA3, this is really an open loop system. We didn't consider feedback. We also made prediction that which is don't have time to talk about here. When we change the EI balance, excitatory inhibitory balance, for example, with the insufficient I to E inhibition, we see that there's a planar tramway would diminish, right? We see the very robust planar tramway diminish. Instead, we see this inter-ischial epileptic discharge line, IED line, spatiotemporal pattern, because with the actual excitation in the circuit, you actually can see this sort of seizure-like, so the IED-like pattern emergence. Although we only focus on the open loop system, but we believe, based on our recent work, the other modeling work, we believe that if we introduce the full circuit as a full closed loop system like MEC, dentate driver CA1, CA3, so with a proper feedback, so we have some reason to believe that this phenomenon, similar phenomenon, can be observed, but this remains to be investigated. So the follow-up questions is that, we talk about tramway in the hippocampus, MEC is really like planar tramway, right? So we see in the cortex, the more complex tramway has been reported. So one of the questions that are we able to generate more complex tramway pattern? And then can we go beyond beyond two brain circuit region, like go beyond the whole brain, right? Can we generate tramway pattern in the whole brain? And then if we have that model, what kind of prediction the model can provide, right? This is sort of follow-up question and natural question people like to ask, right? So in one of the early work in collaboration with a group in Popper School in Johns Hopkins, we showed that by proper, this is a talk about thalamocortical system. So this is a thalamocortical system is well known for the closed loop, right? So we have the cortex, the thalamocortical loop. So we showed that in that world, there's a closed loop system. By introducing the feedback and delay, can really change and can really reshape the tramway pattern based on the assumption you made, in delay and so on. This can really generate very rich dynamic. So just show you one of the video here is essentially showing that cortex, you can actually generate sort of spiral weight, right? And because of the introduction of the feedback and delay. There's many pattern different kind of pattern you can generate. You can generate planar way, you can generate a circular way, or other way. Depends on the some of the parameter we made in the model. And I also borrowed the slide from a postdoc, Guanhua slide. His generalist shared his slide with me. He's doing some of the very large scale whole brain by physical modeling tramway. In his model, obviously, it's very large scale. Using 300,000 single compartment hustling tidal neuron, impose a cell type specificity. Also impose about this connectivity using the Allen Brain Atlas information. Cell type information can be taken from this transatomic sort of data set. So, and this model is highly complex. Obviously, with this kind of model, you can look at more detail, more finer temporal resolution, finer spatial resolution. In this case, he's doing this sort of evolved response, looking at channel wave response to sensory stimuli. Different sensory stimuli could be visual stimuli, it could be auditory stimuli. So just play the video. You see that how does it compare between experimental data and as well as the modeling. So you can see that qualitatively, this model can capture this like a global sort of temporal, spatial temporal propagation structure, right? And really, the cell type information and connectome information will provide a constraint to the model. Okay, so I think I would just basically like to close the talk with some discussion and outstanding question. So what we talk about so far is really motivated by the literature, the phenomenon, and then we do modeling analysis, right? The modeling, however, most of the model, to my knowledge at least, mostly only investigate the spontaneous sort of wave structure, like during sleep, head fix structure, and all passive. Like by passive is like, head fix animal, you have the evolved response. So you present the stimuli, auditory or visual, you measure the brain response, right? And this is more like passive behavior without so much about the active, so the task performance, right? Is especially in the animal world. In the human, obviously you have the memory, like a memory recall, so you can quantify the behavior by, correct or successful, unsuccessful. But in the rodent literature, it's more limited in a sense that you have most of the studies focus on more passive or spontaneous activity. So this raises the questions that whether traveling waves really can we investigate the relationship, the traveling wave causal relationship of traveling wave respect to the behavior, right? Task behavior. How well you can do, is it quantifiable, right? And so far, most of the study, the conclusion is the traveling wave is mostly correlational, so we cannot really I understand the causal law, which one's cause which, a chicken and egg issue, right? It's not causal. So, the only way to look into that question is that through perturbation, right? So you can, recently there's some work has been published suggesting that in human, for example, you can use in transcranial magnetic stimulation to perturb the brain. And meanwhile, you can look in at how does it affect the traveling wave, how does it modulate the neural excitability. So there's a new paper published in Cell Report last year, so to talk about the direction. But however, this task is still limited because the behavior cell is still hard to quantify, right? So there's no quantifiable behavior. You can correlate it, traveling property, a relationship to, for example, particular task, memory task, or any other task, attention task, right? So I believe that, because there's a limitation, so meanwhile, we hope for, a more advanced technique that allow us to experimentally manipulate the train wave pattern to see the causal relationship to past behavior. So modeling probably will, in the meanwhile, can help to answer certain questions. If we can design a task and looking at the train wave pattern and look into the model, see whether we can perturb the model to and looking at the relationship between particular train wave property and then how does it relate to the readout of the behavior. And that I believe will be a interesting research direction to pursue, right? I will stop here and then just acknowledgement the funding resource and then open for some question. Thank you.
Thank you, Sage. That was great.
Thank you. That was awesome. Everyone, please feel free to put any questions in the chat or come up and just ask.
Maybe I'll start. I had a question about you showed a slide at the end of the first half
Yeah.
about entorhinal parahippocampal connectivity driving the hippocampus. And I was wondering if you could elaborate on that result, if you can, maybe you can pull up that slide and I can be more
Yeah.
specific in what
Yeah.
I'm trying to say. But
Okay. Let me go back.
But I'm trying to understand how that result relates to traveling waves. Are there traveling waves in entorhinal cortex, and how do traveling waves relate to the measure that you're talking about of was it functional connectivity? I'm forgetting the exact term that was used for the analysis there.
Is it
Right
this
here.
one?
Yeah,
Yeah.
a Granger causality. That's what I'm talking
The
about. I
Granger
was wondering.
causality. So
Um,
in the Granger causality, essentially talk about whether there's a consistent pattern, right? So if you think about it, the two channel wave are coordinated systematically. So presumably, if they are both strongly theta modulated, so you will imagine that the channel wave pattern, the information from within some, at least within some selected channel, because we randomly select the channel. It's not just based on one pair. And then this should have some strong relationship in terms of predictability if the pattern are coordinated, right, in terms of phase shift, right, because it's the sinusoidal. If you band pass in the theta region, so essentially if there's a consistent temporal relationship, that would suggest that there's a strong so the input output, right? So we found that in the this direction, this information flow direction asymmetric. So there's a stronger predictability, and so the way they interpret the Granger causality, predictability in temporal domain of the frequency is like within this broad range. You can see there's a broad range. There's a one direction is stronger than the other.
but
So
are these traveling waves in the parahippocampal cortex and cortex? Is that what you're talking about here, or is it just a more general phenomenon? I'm just asking what's being plotted here.
this one is not limited to the tramway. The
Okay.
reason, as I said earlier, we don't want to bias in the detection of the tramway to make the argument. So to make the argument, we actually say this is beyond tramway, but probably tramway will make it more pronounced, but the tramway detection is the issue. So in order to increase in the sample size, the detection
Okay.
ability, so we
Yeah.
actually include most of the data that without detecting tramway. So it seems to be I mean, it's a sample size issue.
I see. So this result is measured with Granger causality, but your thought is that this is actually a traveling wave.
There could be a tramway. We're not able to exclusively exclude, right? So one way or the other, we're not able to answer that. So that's why we kind of want to go into the modeling question. The second part of the talk is that we model hippocampal entorhinal cortex. We can see there's a coordinated tramway, at least in by playing the parameter set up. We can see whether how the coordination change if we are able to we can ask similar question.
Okay.
I
Yeah,
mean,
I mean the reason
there's still some
that's
gap
helpful.
there. There's still some gap there. For example, I think the relationship of the hippocampal entorhinal cortex here, I think the exact location could matter. So we don't really know whether it's a medial entorhinal cortex or the exact anatomy could change the story. So our interpretation in here that the human data is restricted because like we are not able to exclude the possibility that there could be subject variability in this implant or other thing.
Well, I
So the
think
one
no, I mean
way,
I think your approach here is sensible. I just think one general thing that I think is interesting is sometimes people use methods like Granger causality, which is a very general tool. Other times people use can measure them and call them traveling waves, but something that's a traveling wave could also be measured with Granger causality to show a significant effect here. So it's not always clear if it's uh, the methods don't perfectly line up with each other. Sometimes they overlap, right?
yeah, yeah, yeah.
Yeah. Okay. I will be quiet now. There's a bunch of questions in the chat. Um,
Yeah, so I actually reading one. Is someone want to coordinate or just read it?
Yeah, so there's a question from Yonan. I'll
Yeah,
put you up to ask in person
yeah.
or in person.
Dentate spine cross region charm begin.
You should be able to speak now if you'd like.
Young, right? That during hippocampal, if I want to read it.
yeah, go ahead and read it. It's
Yeah.
okay.
Yeah,
Yeah.
the question was during hippocampal shower repo or dentate spine, has cross-region charming wave activity been observed? If so, could such wave actually gate which experience a selective and consolidation? This is an excellent question. I don't have the answer for it. Because I mean, to answer the hippocampal shower repo, you need to look at electrophysiology. Probably you need to detect, first of all, the shower repo itself. So I believe that in Kahana's group is I don't know whether it's possible, it's limited because most of the data is recorded during wake. So there's no really sort of like, equivalent to rodents or the sleep or a quiet wakefulness state. To my knowledge, they haven't been reported detection of Schauber ripple. So I don't know the answer for that question. But I think Buzaki's lab probably they might have the data to answer that question, but I don't know this. I don't know the answer myself. Yeah.
Yeah.
Um,
Really great question. I can read
yeah.
the second one
Yeah.
for you. Can traveling waves be compared to sound waves resulting from a series of cars bumping into each other, with the bumps themselves compared to signals traveling down synapses? So traveling waves could travel faster than synaptic waves.
Yeah. Yeah, it's an interesting sort of thought. So I think, I mean, the analogy, I don't know whether it depends on the what time scale. I think I mentioned it early on that in the talk. So it depends on you talk about first order, second order. First order tramway is really limited to sort of synaptic connectivity between neighboring cell. So that's why you have this like axon delay, and then this talk about the oscillator, right? The early word the bar and any other talk about this the delay oscillator. So this is sort of supporting that sort of assumption. But I believe there's other kind of a high order tramway wave phenomenon that can emerge between sort of a feedback system and feed-forward system. You can see this top-down propagation and bottom-up propagation. You do see different tramway pattern, and I think this can emerge. This tramway pattern can emerge with more sort of more sort of coupling and other sort of phenomena. So they had to introduce the closed loop. So I think most of the modeling so far, is a little bit simplified because they, just see whether whether it's input driven or internal driven, so the spontaneous activity. But if you try to looking at the feedback system, even for the whole brain, right? So you have to introduce a time as a delay. Where was the sort of starting as a sort of input? We started tramway start from where and then ends on where, and then whether that whether there's a proper delay feeding back to the input. So I think that require a little bit more sort of investigating in detail based on the exact sort of circuit structure, right? We talk about hippocampal entorhinal cortex here is a perfect example because there's a trisynaptic loop between hippocampus. Also, there's a bidirectional connectivity between entorhinal cortex and hippocampus. I think this circuit is very specialized. Also, we talk about thalamocortical circuit. Also, there's a well-known loop structure. I believe that from our experiences that the modeling, our modeling experiences that by changing any assumption about the feedback or the delay, the traveling pattern can change quite dramatically. Also depends on whether the dominance of the role the tramway is inhibitory connectivity. We always need to introduce a long range connectivity. So whether it's an inhibitory synapse or excitatory synapse. In general, what we found the inhibitory synapse is more robust, and then the excitatory synapse. So I think to answer those questions, I think it's required comparing the literature to see whether the assumption is valid, but also to test the model assumption and test the model robustness to see whether those phenomena we observe in a really sort of robust. And most importantly is to try to make sort of new prediction, right? So some of the data I talk about here in hippocampus part is essentially when we did that work, we didn't really have the right sort of comparison to compare like coherence part. So and later on, when we had this paper, people published independently, then we say, okay, our model can actually make prediction to see whether without fine-tune the model, we didn't really change the model. The model is as then it is, and we just run the model to run the comparison in the coherence analysis. It seems that surprisingly, it actually matched the data well. So I think those things is going to be a loop. So I think the model part is not going to be, it's a require iteration, right? You might require modification of the model or the model assumption.
Okay.
I don't know whether kind of hand waving a little bit didn't answer the question directly.
Oh, no worries. There's also a comment from Bard on the same area.
Yeah,
Bard, if
yeah,
you want
yeah.
to jump in, you can.
Let me see.
I just said that phase waves, which is what these oscillatory waves are, can have infinite velocity. So,
Oh,
because they can be synchronized. If they're synchronized, then it's infinite velocity, right? There's
yeah,
no time lag. So,
yeah,
Bart, I almost feel like the term velocity is misleading. I mean, it
yeah,
appears to move, but there's no velocity implies a physical thing moving, and I think it's distracting terminology. It confuses
I,
me.
I think we've had a lot of these arguments in papers that we've tried to write.
yeah, yeah. I mean, the speed of the relationship is only meaningful when you have a physical distance, right? You're talking about the phase shift between one axis to the axis, maybe like how long is the, introduce the physical distance, and then looking at the relative phase shift, particular frequency, and then you kind of introduce a relative speed. I mean, in a sense, right? Um, yeah.
Okay. Um, I'll move on to the next question from Ganendra. You can unmute and
Yes. Yeah.
ask.
Hi, really interesting talk. I was wondering, you mentioned, does the frequency changes if there is a cross-frequency coupling between, let's say, theta or low gamma? So do you observe these changes in the frequencies of traveling waves, let's say, across hippocampus and parahippocampus, or medial internal cortex or hippocampus, or also across different regions of the hippocampus you showed in one of the slides? And
Yeah.
do you,
So,
yeah.
yeah, I think when we talk about tramway, when we talk about tramway, we looking at the when tramway happen and or without detecting tramway, it's about detection issue. So the tramway, when we detect the tramway, at least we can say the relative coupling is stronger. That's sort of consistent to the literature, whether it's a theta amplitude coupling or the coherence and so on. There's a functional coupling, I think, generally stronger when we detect the tramway. You're saying that we see, and there's no sleep in the data set. There's no sleep phase in the data set. It's all during the memory. So the tasks, I think we are not able to answer the sleep question that you asked. So some of the question that I think you asked is that we are able to look into example, but we're not able to pose statistic because the sample size vary. There's no enough statistical power to say, statistically meaningful conclusion. But we have example. We have some like nice example to show. So like in terms of snapshot. But I think those are whether it's kind of a representative or it's actually outlier, we don't know. So we don't, require more investigation. Another thing we initially want to try is actually in coordination with the finding of the Josh's finding of the cortical tramway, to say what if we can find some subset of the subject that simultaneously you can see the cortical tramway and look into the hippocampal or parahippocampal tramway to see whether those tramway pattern have any sort of a temporal relationship or to make some story out of it. I think there's still I tried to convince the student to look into it, but he didn't have time before he move on.
Yeah, you're right. I mean, also just to follow, because you mentioned that the spectral power is reduced during this
Yeah,
efficient encoding or storage of memory.
yeah, yeah,
So
yeah,
even though let's say the physical distance is fixed, but let's say if these components are operating at, let's say, at criticality, so even though that the structure, the functionally the distance can increase. So are you suggesting from that, let's say, from if we look from the theory of criticality, uh, we observe efficient optimal information processing, and that's why the power is reduced at those specific, let's say, in your spatiotemporal regions. So any comments on that?
yeah. So interesting sort of a direction or question you want to you think about. But I think there's a sort of independent method if you want to go into that. There's a paper that Nature Neuroscience paper. They look into insular hippocampus. So insular is not the parahippocampal, but it's like there's a direct connectivity in the to the hippocampus. during the memory encoding task, similar memory encoding task in human as well, the way they're looking at this data is that they don't looking at channel weight, but they're looking at the change of the insular, especially insular structure, looking at the sort of exponent. So they fit the power. Power basically they have an aperiodic signal. They can characterize the power law. So the power spectrum have the power law. They're looking at the slow, whether it's a steeper or flatter. So they interpret the slow of basically the power law exponent. You fit the LP data with the power law, then extract the exponent. They look at the change of the exponent at different kind of a memory task, whether it's a successful, and so on. So they found that some interesting relationship. I haven't really tried to completely digest the whole message yet, but they found some interesting structures that basically steeper, where there's a steep, it also depends on different region, the insular structure. Some region are more coordinated with the memory task, some region doesn't. Some region don't, different range of the correlation with the task. But generally, there's a exponent, the strength of the exponent is related to sort of excitation. So the EI imbalance in a way. So you can interpret that during so that this aperiodic behavior, aperiodic power spectrum, you can looking at the sort of global cortical EI imbalance related to the memory. So it's not directly, I think, related to your criticality, if I understand correct. Maybe there's no direct connection, but I think there's a different angle to interpret the data is that whether, for example, if you detect channel wave in the insular, there's some structure in the channel wave can distinguish the task, memory performance. So what was the relationship to the hippocampus? And then, I think that they require different perspective to look into it, because some of the data you might not be able to detect the channel wave, but you can use in the finding to sort of interpret your channel wave result.
Right. Thank you so much.
Yeah.
This has been a wonderful conversation, but I think it is time for us to wrap up given the
Yeah.
time. I would encourage any of you who have more questions about Sage's work to reach out to him directly. This is very cool, and I really enjoyed the talk. So thank you so much for joining us, Sage.
Thank you so much. Thanks, everybody.
Please join us next month. This for anyone who's curious. There's Sage's email, which is wonderful. And let's all be in touch. Hope to see you here next month. Take care, everybody. Have a good day. Thanks
Thank
for
you.
joining us.
Thanks
Thanks
everyone.
again for your great presentation, Sage.
Yeah.
Talk to you
Bye.
soon.